Welcome, everybody, to this week's edition of the Extracellular Vesicle Club. We have had a week's hiatus here because the International Society for Extracellular Vesicles held a workshop last week in Desenzano del Garda near Brescia, Italy. And the topic of the workshop was massive EVs, so the production of a lot of material, a lot of EV material that can be used potentially in the clinic. And so we canceled last week's session because of this workshop and because I was participating in the workshop. It was, in fact, the first in-person workshop that ISEV was able to hold since March of 2020. But we're quite pleased to be coming back to the Extracellular Vesicle Club this week as a collaboration of ISEV and SOCRATES, so the Society in Singapore for Extracellular Vesicle Studies. And so I'm very grateful to Minh and to Wei Seong Toh, who are both here today and who have helped to organize this session and will be co-moderating it. So I'm going to hand this over to them in just a second, but I'd just like to remind all of you that during the presentation today, please feel free to put your comments and your questions in the chat box. And then at the end of the session, we will allow unmuting so that you can interact with our special guest today. So Wei Seong, I'd like to hand this over to you, and thank you so much for your help and participation today.
Thank you, Ken. Yeah, so this is Wei Seong Toh from National University of Singapore and US. And today is my great pleasure to introduce Giorgia. Professor Giorgia Pastorin is currently an associate professor in the Department of Pharmacy at National University of Singapore. She is also currently serving as an assistant dean for research at the Faculty of Science, as well as the director of the Pharmaceutical Innovation and Research Center at NUS. Her main research interests include production, characterization, and evaluation of cell-derived nanovesicles, CDNs, as novel biocompatible and targeted drug delivery systems. And today, she will be sharing with us the recent advances in the nano-Cell vesicle technologies obtained through the fusion of cell-derived components with synthetic materials that her team has developed. Over to you, Giorgia.
I would really, first of all, thank the organizer for this opportunity to present some of our work that actually started only in 2018. So by no means we are experts in this field, but I'm really looking forward for some constructive feedback so that we can improve our research. So our group is actually focused on drug delivery. And therefore, if we take one step back and we look at the key requirements for an optimum therapeutic effect, so if we deliver drugs, the drugs need to be potent, right? They need to work very well, but very often they have unfavorable physicochemical properties, for example, low solubility or low permeability, and this affects also the pharmacological effect. Or they interact non-specifically with blood proteins, and again, they are not available as we wished at the disease area. And in addition, very often they are promiscuous in nature, and all these contribute to lower safety profile because many drugs are associated with side effects. Now, nanomedicine is the field, the branch of medicine that utilizes nanotechnology to, for example, prevent or even to treat a disease. And this nanomedicine include mainly for drug delivery, an active molecule, a drug, for example, and a nano carrier. When I talk about nano, I refer to really 10 to the power of minus 9 meters, which is, just to give you a perspective, in the range of viruses or DNA, but it's smaller than conventional cells or even bacteria. And just to give you an idea, if you take one of your hair shaft, it's already 1,000 times bigger than the nanoscale. So I would like to explain a little bit why we need to be at this scale for drug delivery purposes. So carriers, this nanomedicine, are able to carry active molecule to impart some selectivity and improve the safety profile. Now, I was mentioning that we need to have several nanomaterials that can be employed as carrier for these active molecules. And so, for example, we have nanotubes, we have metal particles, polymeric-based micelles, dendrimers, or even lipid-based particles. Now, the question is that these materials have also different properties in terms of size, in terms of surface properties. So for example, you have the polyethylene glycol here, polymeric chain is an hydrophilic chain that is used to decorate the surface of nanoparticles so that it becomes long circulated in the bloodstream. And also they might have different shapes. So there are quite a few groups in the world. So for example, the Mitra Guthrie group in California, that they are looking at particles that have the same material, similar size, but differing shape, and how the shape actually interacts with different biological entities. Now, this nanomedicine, again, the combination of actives with the carrier usually are injected. And therefore, when they start traveling in the bloodstream, when they reach the disease area, a phenomenon occurs. Now, it's a very simplified version, but basically what happens is that in a normal tissue, you have that the blood vessels are very intact, uniform, and the eventual fenestration here between cells are quite small. We are talking about 5 to maximum 10 nanometers. But in case of inflammation, for example, or even tumor, there is a defective vasculature. That means we talk about leaky vasculature that creates fenestration that are even up to 1 micron or even 1.2 micron. And this becomes an opportunity for drug delivery for the simple reason that if here you have your drug imaging this particle that is grayish in color, as you can see, can extravasate both in the normal tissues as well as the disease area. And this accumulating here, you can understand, causes also the some side effects. But nanoparticle and nanomedicine relies on the principle that these particles are too big to extravasate here, and they will accumulate, they will reach the disease area. And this, associated also with a deficient lymphatic drainage, will cause the accumulation of nanoparticle at the disease, and is called enhanced permeability and retention effect, or EPR effect in brief, also called passive targeting, for the simple reason that you don't need to actively modify your nanoparticle. You just need to have a nanoparticle that is of the right size. So we understood that the lower point cutoff is 5 to 10 nanometers, but why we need to still be at the nanoscale, since the fenestration here can be up to 1 micron or even more, is that some reasons that are quite obvious. This because smaller dimension of particles enable to get access to sites that otherwise wouldn't be accessible to bigger carriers. Then carriers at the nanoscales are not able to freely leave the blood circulation, as I showed you before, unless there is this APR effect. And more importantly, even if they are within, let's say, 300 nanometers, they are not easily recognized and removed by the mononuclear phagocyte system, the MPS system. And therefore, they remain in the circulation sufficiently to then reach the disease area. And there are quite a few, I would say, successful stories. This is one of our collaborators, Dr. John Wei Wong and Dr. Bertrand Zerny, that indeed utilized the synthetic nanoparticle called liposome to, in their case, is a myocardial infarction model. And what they saw that indeed their formulation accumulated specifically, as you can see, at the infarcted area. And as indicated also here by further labeling, and they utilize this liposome that are basically a lipid bilayer nanoparticles that is further decorated with the polyethylene glycol, so they are long circulating. And in their case, they were able to deliver an anti-inflammatory agent that decreased the infarcted area within 24 hours, but also improve the heart function. And there are many other successful stories. One, for example, is this Doxil. Doxil actually also is a liposomal formulation with this lipid bilayer that was inspired by nature, in the sense that if you see this lipid bilayer resemble a little bit cellular lipid bilayer, but it's a much simplified and a smaller system. The purpose here was, rather than really increase the pharmacological effect, was really to decrease side effects, because doxorubicin, the drug that was carried, is highly cardiotoxic. By incorporating it in a nano formulation, it basically decreased these side effects. So despite these very successful stories, we also have some additional limitations, I would say. They still need to be solved. The first thing is that even if the nanoparticles are able to reach the disease area, they are not necessarily uptaken efficiently by target cells. In addition, they are cleared by the MPS system, as I said, unless they are further decorated with this polyethylene glycol. Or still we see that we are assisted at the accelerated blood clearance phenomenon, for which when you inject 2, 3, or multiple times, all in a sudden the concentration in the bloodstream actually drops. And last but not least, there are still some issues of immunogenicity, such as this kappa effect or complement activation related pseudoallergy that is basically unpredictable. It's not necessarily dose related, but it can be life-threatening. And this has propelled scientists really to think maybe we should go back into nature and consider what is nature providing. And indeed, what we have here is that we know that cells secretes their own drug delivery systems. In fact, if you look at this vesicle, in particular, the smaller extracellular vesicles, they are released, they carry important information, they are uptaken efficiently by both neighboring as well as distant cells, and therefore they are recognized. And all these make this system ideal for drug delivery. In particular, you have that extracellular vesicles are biocompatible, and because they are total nature in nature, they can be at the nanoscale, especially if they are small in size. They can reach the disease area. Why not? By exploiting the EPR effect that I mentioned before, similar to syntactic nanoparticles. They have shown the ability to cross also biological barriers, and we have summarized this in a recent book chapter. And this is quite unique, I would say, of these extracellular vesicles. They have intrinsic targeting ability. What I mean by that is that they preserve some surface cues from the parent cells that enable them to be recognized and uptaken very efficiently by target cells. And last but not least, in principle, they can be loaded with active molecules to then work really as a proper nanomedicine. But this I put here as is still suboptimal for the simple reason that these are bags that are not empty, and therefore they're already full of precious material, and trying to incorporate further exogenous material is actually still a little bit challenging. And therefore is where we found ourselves in front of sort of dilemma. Should we continue working with synthetic nanoparticles that have established protocols that have been used even clinically? You can control very well the size, the polydispersity, the functionalities on the surface. Or should we try to instead exploit the intrinsic targeting ability of the extracellular vesicles? On one side, we would need to deal with the immunogenicity or the kappa effect that I mentioned before. But in case of extracellular vesicle, we still have limitations in terms of drug loading and eventually scalability. It seems that scalability now, according to Kenneth mentioning early on, is that maybe has been overcome. But in terms of drug loading, still is a little bit challenging. And this brought us to conceive a new system, a biohybrid system that is derived from the fusion of a conventional drug delivery system and extracellular vesicle or cellular components. Now you can understand that with this system, we try to get the best of two worlds. We are able to load the drug without any problem. We can functionalize eventually the surface, but at the same time, we take advantage of the intrinsic targeting ability eventually and improve biocompatibility of this whole system. Now, despite this, I would say, very enthusiastic slide, we need also to recognize that the formation of this system requires an additional steps actually in the formulation process. And we are really need to work still a lot, especially in terms of characterization of this system before really can be brought into a more translational level. So first of all, how do we obtain this system? We take basically cells, and by exposing them to hypotonic conditions, so we create transient pores that enable the removal of the intracellular component. And then by exposing to physiological condition, you have the closure back of the membrane in the form of the so-called cell ghosts. Now, we demonstrated that you can remove quite a lot of the intracellular content, even the nucleus. We saw here that the nuclear content is much more scattered. And once we look at the concentration, the DNA content, we saw that there is a drastic reduction from the cell to these cell ghosts. And even more when we finalize our biohybrid system that we called nano-Cell Vesicle Technology, or nCVT in brief. And then a further proof of the emptying process was with this green cell tracker that is becoming impermeable when enters the cells and interacts with the thiol groups of intracellular proteins. And we saw that according to basically the osmotic conditions that we utilize was really the driving force for the emptying of the intracellular content. And these cell ghosts, we proven that are non-viable in nature because they couldn't really expel the trypan blue staining, and also they were non-proliferative in nature in comparison eventually to the U937 monocytes that we use as a prototype. Now, I want to specify that even though we use this as a proof of concept, this type of protocol almost with no modification can be applied to any type of cells. So we have these cell ghosts, and then the next step that we did was simply to hydrate a thin film of lipids with an aqueous solution containing the cell ghosts. Now, because lipids are not loving water, what they tend to do, they tend to close up, forming these multi-lamellar vesicles that incorporate the cell ghost. And then by extruding them, we obtain this final nano-cell vesicle technology biohybrid system that resemble a little bit the liposome because it has a lipid bilayer, but it also resembles a little bit the cellular component because it's supposed to preserve some proteins and lipids from the parent cells. And then according to the physicochemical properties of your drug, you can load the drug after this vesicle has been formed or during also the preparation steps. But then you can deliver to target cells and then further perform some studies. In terms of physical characterization, we proved that these nCVTs are actually similar inside to the synthetic counterpart, the liposomes, and they are also similar in terms of particle concentration as determined by the nanocytes. We demonstrated that the fusion is really a fusion what occurs between the two entities, and we demonstrated this to a FRET analysis whereby we had two fluorophores, as indicated here, associated with the lipids, and they were given a signal here, indicated in yellow. In the moment in which there was the intercalation of a cellular component, the two fluorophores were apart one from each other, and we saw a dose-dependent drop of the signal. Another proof was through immunoprecipitation with these Dynabits with streptavidin that were basically able to complex with lipids associated with biotin. So both the nCVT and the liposome were able to bind these bits, but only the cellular component that was labeled with CY5.5 showed also this additional signal. And therefore, we found that our nCVT displayed both the signal for the liposome, so the lipid content, as well as the cellular content. In terms of biological characterization, we assisted to a remarkable drop of protein contents. And mainly we did a proteomic analysis, and we saw that the proteins that were here were mainly membrane protein. And this is not surprising because we have removed all the intracellular, almost more than 75% of the intracellular content. And we found that some examples of exosome markers still were present in our nCVTs, and these are also known to be present in some cells such as B or T cells or even on our monocytes. But another hypothesis that we had is that if our hybrid system contains both the cellular component as well as the lipid component and derives from cells, parent cells that contain some molecules that are preserved also from the cells to the cell ghost and then into the hybrid, there could be some opportunity for us to see whether there are some molecules that can favor some targeting, some selectivity or specificity towards some target cells. And one of such interaction is represented by endothelial cells that in case of inflammation or tumor, they tend to overexpress cell adhesion molecules or CAMs. And these CAMs tend to interact with the partner integrin or selectin that are present on the monocytes. Now, the question is, are these present still in our nCVT? And in this case, we observed that indeed there was a preservation from the cell to the cell ghost, and also in our nCVT of this CD11A, which is a part of the lymphocyte function-associated antigen 1 that is a receptor of the ICAM. So this was a proof that there was some preservation of key proteins from the parent cells, but we wanted also to understand whether this was also involved in the uptake of the nCVT inside the endothelial cells. And to do that, we took UVEC cells and then we treated them with the TNF inflammatory cytokines in order to kind of mimic what happened at the inflammatory site. And for that, there is an overexpression of this ICAM, VCAM, or E-selectin. And we wanted to see whether would this situation improve the cellular uptake of our nCVT. So when you consider liposome that were basically our form of comparison, there was no much of a difference with or without the TNF alpha. But in case of our nCVT, as you can see, there was a remarkable increase in the upon treatment with TNF alpha, indicated that indeed this also interaction mediated, at least partially, the uptake, which was almost double of nCVTs. And these were then brought back to the basal level when we treated this sample with an antibody cocktail containing anti-ICAM, VCAM, and E-selectin antibodies. And this indicates and confirms that in principle, we can take advantage of this interaction that causes an intrinsic sort of heterotypic targeting. In terms of drug delivery, we actually loaded quite a few types of molecules, big molecules like albumin, small molecules like this one that is doxorubicin, or antisense oligonucleotides. And we are trying to see also, is there a limit in the loading efficiency? But apparently, because we are really leveraging on what has been already established for liposomes, we can get 80% or even more of loading efficiency. In this specific case, we just exploited the fact that the doxorubicin has a basic functional group here. So if you utilize different pH gradients, you can favor the diffusion of the doxorubicin through the nCVT. And because inside here is slightly more acidic than outside, your molecule becomes protonated, it precipitates out and is unable basically to come out. And this provides a lot of stability to our sample. Sample that still proved we have, even depending on the cell type, even more remarkable differences in the uptake. But meaning to say there is even if we have a small amount of proteins eventually or cellular component that is conjugated to 2 milligrams of lipids, still it makes a difference in the cellular uptake. And then we had a small proof of concept in a colon carcinoma model, whereby we first injected subcutaneously some CT26 colocarcinoma cells. We waited for the tumor about 2 weeks to develop, and then we tail vein injected our samples containing the doxorubicin. And then we monitor and we sacrifice the animals within 20 days. This protocol is still not optimized. We had to accelerate that due to our imminent lockdown, but still you can see that there was a difference. Our nCVT still provides some improvement in terms of pharmacological profile, and also without a noticeable systemic toxicity due to the lack of drop of weight in mice. And this was the difference, not just with the control, but also with the liposomal formulation. Now, what was interesting in terms of systemic toxicity, we also analyzed some parameters, let's say in the heart, in the liver, in the kidney, in the bone marrow, in the thymus, in the, and so on and so forth, to look for eventual toxicity. But consistently, our nCVT showed that there was lower cytotoxicity, not just to the cardiotoxic doxorubicin, but also in comparison, for example, to the liposome, suggesting that if we can improve slightly the pharmacological effect, in every case, we gain quite a lot in terms of decreased side effects. And in the last few minutes, I would like also to show another proof of concept that was tried really to help us to understand a little bit better our cell vesicle technology system. This in terms of transfection efficiency. So in case of transfection, the idea is eventually to incorporate an exogenous DNA, for example, into cells. And for that, you need also, there are different ways of doing. Viral methods are notoriously very efficient, but also they have the drawback of safety. Other methods, such as physical and chemical methods, are those that are adopted more frequently in the laboratory. So, for example, lipofectamine is one of the most utilized transfection reagent in the labs, and it consists of a nano formulation. Actually is lipid-based nano formulation consisting of positively charged cationic lipids. And basically why is that important is because this positively charged lipid then interact with negatively charged plasmid, which otherwise will not be able to enter the cells, and therefore the complex is transferred inside the cells so that they can express a certain gene of interest, and then they can be analyzed. This is the common scenario, and these are also the most widely utilized cells for the transfection. They work very well. The transfection can be complete 80, 90, 100%, but it's not true for all the cells, because there are notoriously some cells that are called hard to transfect, HTT cells, that includes, for example, primary cells, stem cells, even the U937 cells that I was mentioning before, the pre-adipocytes. They really are much more sensitive to the reagents and to the treatment that are used for transfection. And very often they are associated with such a high cytotoxicity that the transfection efficiency is quite low. And therefore, we wanted to see, since lipofectamine is a liposomal formulation, is there any advantage of using our cell vesicle technology? So we used the same similar approach that I showed you before. Simply, we replaced the lipid component with a positively charged lipid in order to complex with a plasmid for the GFP expression. And then this complex was incubated in hard to transfect cells. In particular, we used 3T3L1 pre-adipocytes. Now, what was interesting to observe and gave confidence to us is that it was important to use CVTs and nothing else than that, because if we use, for example, only cell ghosts, so only the cell membrane, as you can see, it is highly negatively charged, and therefore was not efficient in complexes with the negatively charged plasmid. And the same is applicable also if you use just cells that are complex with the lipids. The content in cells is far beyond the positive charge of the lipids that you add. So only when you have really the CVTs or even positively charged liposomes, you can have a net positive charge that then nicely complex with the negatively charged plasmid. And we could even modulate how much plasmid we could really incorporate into these CVTs. So not surprisingly, in terms of transfection efficiency, as I said, cell ghost of cells were not able to complex with the plasmid, so there was no transfection. In the case of the cell vesicle technology, it shows an improvement in the transfection. I wouldn't say it is a spectacular improvement. It is quite remarkable considering the harshness of these cells to be transfected, was better than the liposome, but for us was important to see that we had a remarkable gain in terms of cell viability. As you can see, the dotted liposome might be able to transfect 15, almost 20%, but with a lot of drastic decrease in cell viability. And that was not the case for our cell vesicle technology. And this is true also when we use lipofectamine 3000, the gold standard for the chemical transfection, whereby you can see that we could reach about 15% of transfection, but this value is actually the maximum that we could utilize, because then we had a lot of cytotoxicity. And instead, in case of our Cvts, we could continue actually with the transfection. Only at a certain point, we start seeing that the cytotoxicity emerged. So this was the first kind of part that we wanted to prove. And another question was, does it matter the cell from which we produce the cell vesicle technology? Does the choice of cell impart some selectivity in the transfection? And the answer is yes. As you can see, when we use T3L1 cells to produce the cell vesicle technology, to transfect these cells, it works better than if there is another type of cells that is utilized. And that suggests that, and still the cell viability is actually the same. This suggests really that there is also a homotypic or like-to-like selectivity in terms of transfection efficiency. And this was the second point, that learning point for us, that we understood that according to the cells basically that you choose, then there could be a modulation of the transfection in this case. And last but not least, we wanted to see whether does it work with or without serum? And this because one of the drawbacks of these chemical reagents is that they always need serum-free conditions. So as you can see here, we didn't compromise much, especially for longer time of transfection in terms of transfection efficiency or eventually cell viability. And that indicates that eventually we could adopt this system in a more physiologically relevant context. And so I think with this, I came to the end of my presentation. What I would like to suggest is that these nCVT are a new type of delivery system, and in some instances they can show improved properties in comparison to conventional nanocarriers. They have equal drug loading properties, but then they can be uptaken with so logarithmic scale higher uptake in comparison to conventional carrier. Then according to the cell of origin, it is possible to impart some homotypic or heterotypic targeting, but still a lot has to be done. And then in fact, our future work will include a lot of characterizations, assessment of what is the main mechanism of uptake. Do they behave more like cell ghosts, or do they behave more like liposomes? And also some immunogenicity to assess the immunogenicity of the system. So with this, I would like to thank, of course, the funding that is available, the group, in particular, the students that made most of the work that I presented today, our collaborators, and of course, all of you for your kind attention. Thank you.
Thank you. So thank you for the very nice presentation. So anybody have any questions can just put into the chat box. So maybe I will just start off with some of the questions in the chat box.
Sure.
Yeah. So Lydia was asking, okay, is it possible to apply your technology after EVs were produced by the cells and creating EV ghosts instead of cell growths in order to keep the production?
Yes, the answer is yes. We have tried that. The reason why originally we have different generation, if you want, of this cell vesicle technology. One was really to use the EV and infuse with the lipids. Having a fusion of the EV with the lipids works, but we called it exoplexis. But there is a little bit of an issue of aggregation, which we didn't observe when you empty. If you take the EVs and then you empty, that is not absolutely a problem. So it is possible. What we are worried eventually, if I have to think EV, empty EV, so EV ghosts and fuse with the lipid, or cell ghosts fuse with the lipid, you might have less protein contents. But maybe, you know, you have the key ones that you need.
All right, so the next question. So thank you for sharing the work on the doxorubicin-loaded nCVTs. Do you have any information on the biodistribution of these vesicles and the pharmacokinetics?
Yes, I do. I don't think I have here. But anyway, what we see is that we see an accumulation at the tumor site that actually was higher than the one at the in correspondence with the in comparison with the liposomes. They still go quite remarkably in the kidney, and also in the we found them in the liver. So it actually depends a lot on the time of assessment. So let's say if I look at the biodistribution within 24 hours, we find them in the liver, we find them starting to be in the kidney, but also at the tumor site.
Okay. So another question from Santosh. Any differences in loading for hydrophilic versus hydrophobic versus lipophilic drugs?
That's a good question. And the answer is that in principle, no. The reason for that is that your drug can be loaded during the hydration. If it is water soluble, for example, it can be, maybe if I just can go one moment to the slide, so maybe it's easier to explain. Can you see the slide or?
Yep.
Yeah. Okay. So when you, the drug, if it is not water soluble, you can dissolve this within the lipid here. And this is a process that is done for liposomes, for example. You dissolve the not water soluble drug here. And then when you fuse them with the cellular component, your drug, not water soluble, will be mainly in the lipid bilayer of your nanovesicle. In the case in which it is hydrophilic instead, you can either load it, as I said, that this case we load it after we create this CVT by remote loading, so-called, by playing with different buffers, or you can also load during the hydration step here. The only thing is that when you extrude, you can lose a little bit of your drug that is not encapsulated into the cell vesicle technology. Having said that, one recommendation that I would do to make sure that the drug, especially those that are not water soluble, to remain more stably in the lipid bilayer, is to use not just lipid, but also cholesterol. Cholesterol imparts some stability to your formulation. And that's where, once the formulation is stable, your drug remains stably there.
Okay, so Minh has another question. Okay, so do you use CT26 cells to produce CVTs for CT26 tumor treatment? And does it help to increase the biodistribution of the CVTs to the tumor?
That's an excellent question, Minh, and as always, you have very insightful ones. Not yet. We haven't done that yet. We wanted to do that just before the animal work, and then the student finished her PhD, and therefore was not an opportunity. But that's an interesting question that we can definitely test and check. Thank you.
Okay, so Santosh has another question. So after loading, how do you remove free drugs and purify the EV liposome system?
Yeah, we adopt actually the same methods that are done for the liposome. We can use dialysis, for example, extensive dialysis. You can use also size exclusion chromatography. We can use, we usually combine also different methods according to the physical chemical property of the drug that we want to remove. These are no different from conventional liposomal formulation.
All right. Yeah, so I have a question. So when you are trying to have the fusion, right, to create these CVTs, how do you actually control the materials from the cells that you want to fuse with your liposomes? Are you able to have some form of control for the specific membrane proteins or even any receptors that you wanted to specifically fuse with your liposomes?
I don't have a precise answer to this. What we do is that maybe I forgot to mention that when we create this system is also done in quite viscous condition. We use a 0.06% sucrose, being extremely viscous. That is minimizes the eventual membrane flipping and keeps the integrity of the membrane as much as possible. So this is a mini control that we can have. We are still at the phase where we don't know which component of our final nCVT is eventually working. The idea is actually to take the advantage of the cellular component, try to identify what works, and then through proteomics, lipidomics, we analyzing even microRNA, everything analyzed, try to then have a simplified system that is synthetic eventually in nature and only contains the proteins, for example, or the lipids that we need. So we are still, it makes perfectly sense to your question, but we are still not at the phase where we can exactly say, I preserve this protein or these are the, yeah.
Okay, yeah. So any questions from anyone? Yeah, so there's another, okay. So do you have an estimate of how many EVs produced per cell?
Produced per cell, Ula, is a number that I don't have with me. Let me just what we know is that this is the number of particles that we obtain from one E7 cells. Yeah.
Okay.
So then we should be able to calculate. Yeah.
Hi, Georgia. Relating to that question, I wonder if you can scale up better when you use Cvts compared to when you have EVs alone. So now you have like double amount of materials. So is it better to scale up the production of this particle?
I'm not sure I understood, Minh. What we can do is that we can definitely scale up. We have spent quite some time to see how much we can do. And actually we can do 2-4 milligram. We can utilize according to the extruders that you have. There is no limit in such a thing. What we do is that imagine we start with one at the current state, 1E7, we convert, actually the conversion to cell ghost is pretty quantitative, and then this same amount is complex or fused with 2 milligram of lipids. But this can be scaled up without much of an issue. We saw that, and the behavior, uptake, eventually properties were really preserved. So I don't know if I addressed your question, Minh, but in principle, you can scale up. Yeah.
Yeah, I see. That's cool.
We
think it's
just limited to.
Yeah, I think that is better than if we grow the cell and let them to produce EVs by themselves.
Okay. Yeah.
Yeah. So there's another question coming in. Okay. So do you actually see any randoms of cellular materials in the Cvts?
Random cellular material, like what, for example?
So I suppose I think Ali is asking about apart from the cell membrane, okay, that is being fused with your vesicles, with your liposomes, do you actually see any of these other cytosol materials that also have incorporated into your Cvts?
Well, we were, was it? We still have a little bit, we have something like 25 nanograms of DNA content that we are trying to push to remove even further. So we can remove between up to 75% or slightly more of nuclear content, but you see that there is still a little bit that we need to push and work to minimize as much as possible that content. So if that is an example of still cellular material, nuclear material in this case.
I see. Okay, so another question, is there any differences in size for loaded versus unloaded nCVTs?
It depends a little bit on what system you are looking. For example, is an interesting question. In the case of the transfection that I show you, there is not an increase with once the plasmid is complex, the size is the same. Usually, the size does not increase beyond what is if it is let's say 150 nanometers is really very narrow, the increase with an active. If then, of course, you put a long polyethylene glycol chain around it, then there is an increase of maybe plus 20 nanometers of size, according to how big is your polyethylene glycol that you fuse, that you conjugate.
Yeah. So any other questions coming in? Okay. So another question from Bo Hong, are the compositions of the nCVTs consistent?
Yeah, very good question. Yes, consistent in the sense that when we did proteomics, for example, in replicates, and the majority, especially in the case when we did CVTs from stem cells, imagine that even we did a comparison between adipose-derived stem cells and mesenchymal stem cells, and what we observe is that the difference of the content was absolutely minimal. So in that sense, the system is quite reproducible.
Yeah. So it's a very robust system that you have a very good consistency.
Yes, it gives good consistencies. Yeah.
All right. So thank you very much for the very good presentation. A lot of work that has been done. So we are coming to the end of the hour. Okay, Kenneth, you have anything to add?
Well, I would just like to say as well, thank you very much, Giorgia, for this presentation. I hope that you've gotten some good feedback and perhaps some new opportunities as well. I know I'm going to be reaching out to you because I have a couple of questions, and I think you might be able to help me with those. So this has been really great as an introduction to your work, and we wish you all the best as this moves forward. So thank you for this. Thank you very much, Wei Seong and Minh, for helping with the organization and the moderation today. And thank you to all of our participants and for the excellent questions that have come in. So I hope you all have a good rest of the week, and I look forward to seeing many of you again very soon. So take care, everyone.
All right, take care.
Thank you so much.
Thank you.
And
Thank you.
everyone.
Thank you, everyone.